Hello everyone, welcome back to the channel. Today we will discuss one of the most revolutionary discoveries in modern molecular biology, CRISPR-Cas9, a powerful genome editing tool that has transformed genetic engineering, biotechnology, medicine and agriculture. So consider it as a smart molecular pair of scissors that can find the exact mistake, cut it out and replace it with the correct word or correct sequence. So that's basically what CRISPR-Cas9 does. So the main characters are, first one, guide RNA or gRNA. It's like a Google maps the location and it tells CRISPR exactly where in the DNA to go. Then we have Cas9 protein, the molecular scissor. Once it reaches the correct DNA location, then Cas9 cuts the DNA, and third one, fixing desired DNA sequence is inserted. So let's start in detail. Introduction. So CRISPR is clustered, regularly interspaced short palindromic repeat, and Cas9 is CRISPR associated protein 9, constitute a natural adaptive immune system found in bacteria and archaea to defense against viral bacteriophage infections. Key component: Cas9 protein, endonuclease act like molecular scissor, creating a double stranded break in DNA. Second one, guide RNA. So it's the complex of two RNA, CRRNA and trRNA, CRISPR RNA and transactivating RNA. So in lab, these are fused into a single molecule called sgRNA, synthetic guide RNA. Third one, very important, PAM. So PAM sequence, a short DNA sequence, usually this, immediately following the target DNA sequence. So Cas9 will not cut if PAM sequence is missing. Very important. So the target recognition, Cas9 along with sgRNA scans the genome for PAM sequence. This one, binding and unbinding. Once the PAM sequence is found, Cas9 unwinds the DNA. If the sgRNA matches the target sequence, this sequence, it binds. Third, cleavage. Cas9 uses two nucleases domain, this and this, to cut both the strands of DNA, typically 3 base pair upstream of the PAM sequence. And the last one is joining with or fixing it with desired or correct sequence. The mechanism of action, the process occurs in 3 primary steps. First one, target recognition. Cas9 loaded with sgRNA scans the genome for PAM sequence. Second one, binding unbinding. Once PAM is found, Cas9 unwinds the DNA. If the sg matches with the target sequence, it binds. Third one, cleavage. Cas9 uses two nucleases domain, H and H and RUVC, to cut both the strands of DNA, typically 3 base pair upstream the PAM DNA. Next, DNA repair pathway, the editing part. After the DNA is cut, the cell attempt to repair the break. This is where the actual modification happens. So first one is non-homologous end joining. The cell glues the end back together, results in highly error prone, often causes insertion and deletion, leading to gene knockout or loss of function. Second is homology directed repair. A repair template or donor DNA is provided by the researcher. So we are using it for this purpose. So now gate specific technical points. HNH domain cleaves the DNA strand complementary to the sgRNA. RUVC like domain cleaves the non-complementary non-target strand. Dead Cas-9, a mutated version of Cas-9 that can bind to DNA but cannot cut it. It is used for CRISPR interference or CRISPR activation by fusing it with transcription factor. Off-target effect, a major challenge where the gRNA bind to the similar but incorrect sequence elsewhere in the genome. So quick comparison between CRISPR and the other tools. CRISPR Cas9 or zinc finger nucleases. Target mechanism, it uses gRNA to target DNA sequence, uses engineered zinc finger proteins to target DNA. They are relatively easy, RNA based. Zinc finger nucleases are complex, protein engineering required. CRISPR have high, easy to retarget. They are lower, difficult to redesign. CRISPR Cas9 cost is lower, zinc finger cost is higher. Application in CRISPR Cas9, gene knockout, activation, repression, editing, and in zinc finger nucleases application is gene knockout and correction. Recognition, CRISPR Cas9 can recognize RNA DNA interaction, zinc finger only protein DNA interaction, talents only protein DNA interaction. Ease of use, very easy, difficult, moderate. Cost, low, high, high. Multiplexing, it is there, can target multiple genes, difficult, difficult. Tip, why Cas9 doesn't cut the bacteria's own DNA? Answer is bacterial CRISPR locus lack the PAM sequence. Very important, यहाँ से गेट का क्वेश्चन आया था. Next, let's do some questions. CRISPR Cas9 was originally discovered as bacterial adaptive immune system. Question 2, the Cas9 protein primarily functions as endonucleases. Question number 3, in CRISPR Cas9 gene editing target specificity is determined mainly by guide RNA. Question number 4, which sequence must be present adjacent to the target DNA for space Cas9 activity? So it's NGG sequence. Question number 5, the PAM sequence is recognized directly by, so it is recognized by Cas9 protein. 6th, Cas9 introduces, so it introduces double strand break. Repair of Cas9 induced DNA break by non-homologous end joining generally results in gene knockout or loss of function. Question number 8, homology directed repair requires DNA repair template. 9th, which DNA repair pathway is more error prone? So it is option B, non-homologous end joining. It is more error prone, chances of errors are more in this. Question number 10, a CRISPR experiment aims to delete a gene. Which repair pathway is usually exploited? So non-homologous end joining is usually employed to delete a gene. Next, question number 11, a gRNA guide RNA contains 20 nucleotides complementary to the target DNA. If one nucleotide mismatch occurs near the PAM sequence, the most likely outcome is reduced cleavage efficiency. Next, which of the following is not a component of CRISPR Cas9 editing system? So DNA polymerase 3, this is not the system, this is not the component of the system. Question number 13, The CRISPR locus in DNA, the CRISPR locus in bacteria contain, so it contain repeated sequence separated by spacer. Question number 14. The spacer sequence in CRISPR array originate from, so it originate from invading phages or plasmids. Dead Cas-9 is used for, so dead Cas-9 is used for gene regulation without DNA cutting. Question number 16. A researcher want to replace a mutant allele with a normal allele. Which pathway is preferred? So in this, homology directed pathway is preferred. Question number 18. Which step occurs first during CRISPR mediated genome editing? So in this, guide RNA binds with target sequence is the first step. Question number 18. Off target effect in CRISPR refers to, so it refers to cleavage at unintended genome sites. Question number 19. From Streptococcus pyogenes belong to which class of nucleases? Cas9 from Streptococcus pyogene, so it belongs to RNA guided endonuclease. That's all for today. So see you in the next lecture. So here is some short description or comprised description important for GATE exam. So see you in the next lecture. Till then, bye bye. Please like, share and subscribe.